Yeah,
Okay,
we
excellent.
can hear you well.
So we can start.
Yes.
Okay, really thank you for this kind invitation to present at the Enjoy Science seminar series. It's an honor and pleasure to be here. As you know, most of you have been here for just over a year, and my lab is now up and running. And I thought of discussing a couple of relevant works that we have done over the year regarding the metabolic changes and essentialities in leukemias and lymphomas, and this is in particularly may link to things that we expect to do and hope to do here at MD Anderson with my lab. So as was nicely introduced, I'm working on cancer metabolism. It's been quite a while since we started this process already 25 years ago, while I was a postdoc. And the first questions that came to mind at the time, now it might be a bit more obvious, is why should we link metabolism to cancer? I mean, this was the era of oncogenic drivers and tumor suppressor gene loss and the genetic era of cancer biology. And adding a biochemical metabolic aspect to it required some justification. And I think the best way to describe it is as follows. Cancer, by definition, is a proliferative disorder and therefore require an anabolic process. There is an anabolic demand for building biomass. This is something that every tissue that grow requires, but particularly tumor that grow indefinitely will require deregulated anabolic processes. This means the conversion of nutrients and small molecules into macromolecules in order to make cells, membranes, proteins, DNA, RNA. These are the major biomass component in our cells. At the same time, almost by definition, cancer cells are positioned in a metabolically stressful environment. We know very well about the oxygen limitation and the response to oxygen limitation, but the reality is that it's not just oxygen that tumors are limited with. There's also a problem with nutrient availability in general, and particularly those that diffuse less well, like lipids. So that's something I'm going to touch upon today in my talk. And finally, in many ways, it's actually very interestingly because it connects genetics and metabolic, putting together elements of genetics and metabolics. There are mutations, oncogenic and tumor suppressor mutations in genes that are metabolic enzymes. These include TCA cycle enzymes and or TCA cycle related enzymes. I'm not going to talk about it today, though it is or it was a major focus of my research over the years. So today I'm going to focus on the environmental stress that leukemic cells are exposed to. So before we go deeper into the research, I want to give you a general brief introduction to catabolic and anabolic processes in our cells. Catabolic processes for energy production and anabolic for biomass production. So what you see here is a process of glucose oxidation in the cytosol into pyruvate. This is so-called glycolysis. It's a process that can happen very quickly, assuming glucose is available. And then it produces both NADH as a reducing equivalent for further oxidative phosphorylation, but also ATP in an oxygen-independent manner. Pyruvate then gets into the mitochondria, where it further oxidizes in the TCA cycle, producing more NADH and FADH2, which are then oxidized along the electron transport chain eventually leading to ATP production in the process called oxidative phosphorylation. This is an oxygen-dependent mechanism, as oxygen is the ultimate electron acceptor from NADH, from the reducing equivalents. So this is the catabolic process of glucose all the way to CO2 and water, generating large quantities of ATP. But it's limited because it requires oxygen. So under anaerobic conditions, when oxygen is limited, what cells do in anaerobic metabolism, they produce lactate. Lactate is by and large produced for solely one reason, and it is to recycle NADH back to NAD+. That's something that, people tend to forget from old biochemistry. The reason for it, in order to be able to run glycolysis without oxidizing NADH in the mitochondria. Otherwise, there'll be no glycolysis and there'll be no energy production. So you probably have heard of Otto Warburg, maybe the founder of the cancer metabolism field 100 years ago. Otto Warburg studied electron transport chain. He actually characterized the electron transport chain, and he received a Nobel for that in the 30s. However, he spent much of his career working on cancer research. For reasons that I'm not going to go into, and it's actually quite fascinating. He focused on what we now call aerobic glycolysis, and this is the production of lactate even in the presence of oxygen. So this is how he defined cancer. I'm not going to say that he was right. Probably in many cases he was not. Particularly that his major understanding of the mechanism, or the conclusion that he drew, was that cancer derived from cells with defective mitochondria that cannot do oxidative phosphorylation. And now we know that it's in the very rare cases in which Warburg was correct with this statement. Nevertheless, we do know that cancer cells take a lot of glucose and produce a lot of lactate, even in the presence of oxygen at time. Part of the reason for taking a lot of glucose potentially has to do with anabolic processes, not exactly in line with lactate production, but glucose carbons can be diverted to many pro-anabolic precursors, namely amino acids, as you see here. These are all the non-essential amino acids that our cells capable of producing themselves. And by and large, the carbon source for it is coming from glucose. And they are derived either from glycolytic intermediates or from the TCA cycle, as you can see here. So again, that links glucose metabolism to anabolic processes. Furthermore, probably heard a lot about the pentose phosphate pathway, a way to shuttle glucose or glucose 6-phosphate into an alternative pathway that will produce ribose 5-phosphate, which is essential for nucleotide biosynthesis. Along the way, it can also produce NADPH, an important reducing equivalent that is a key mediator of lipid biosynthesis and managing redox stress. Now, this is only the ribose 5-phosphate and maybe later the oxyribose that will derive from it. But to make nucleotide, you need the nucleobases, which the carbon and nitrogen of these derived, among other sources, from amino acids such as glutamine, glycine, and aspartate. So again, linking all the carbon metabolism of glucose also for nucleotide biosynthesis. So now we have protein and DNA and RNA as macromolecule in that scheme. What we're missing is the lipid component. So for de novo synthesis of lipids, and I'm going to elaborate on that more in my talk, cells need to produce first fatty acids, the long chain acyl groups. For that, they require acetyl-CoA in the cytosol. This process happens in the cytosol. Nevertheless, acetyl-CoA by and large is produced in the mitochondria, and it doesn't cross the mitochondrial membrane. So the way to transfer acetyl-CoA to the cytosol is by synthesizing citrate in the TCA cycle, exporting citrate to the cytosol, and then breaking it down to acetyl-CoA and oxaloacetate. This is an important, a crucial step in fatty acid biosynthesis. And then acetyl-CoA is building the fatty acid with the help of NADPH that I mentioned before, that largely derive from the pentose phosphate pathway. Once fatty acids are produced, they need to be esterified into glycerol or phosphoglycerol to make triglyceride or phospholipids, etc. This derived from glyceraldehyde 3-phosphate directly from the glycolysis. So again, you can see the link between all the catabolic processes and the anabolic processes. The one thing that I want to highlight here, because I'm going to touch upon it a little bit later, is that the TCA cycle, by and large, is carbon neutral, meaning that it's a circle. It doesn't produce really extra carbon. It gets 2 carbon from acetyl group here, and it loses them to CO2 along the dehydrogenase, the isocitrate dehydrogenase and alpha-ketoglutarate dehydrogenase processes. So basically, you start with oxaloacetate, which is a 4-carbon molecule, and you finish with oxaloacetate, which is a 4-carbon molecule. So there is no net gain of mass by the TCA cycle. What it means that if we start depleting the TCA cycle to make amino acids or fatty acids or nucleotides, then we're going to lose the energy component. We're going to lose component of the TCA cycle. Therefore, there is a constant need to replenish the TCA cycle molecules. This is called anaplerosis. The major anaplerosis enzyme in our body is an enzyme called pyruvate carboxylase that actually add a carbon from bicarbonate to the 3-carbon molecule pyruvate to make the 4-carbon molecule oxaloacetate. So this way now you contribute net 4 carbons to the TCA cycle through pyruvate carboxylase. Another process which you may be more familiar with is the glutaminolysis, the breakdown of glutamine into alpha-ketoglutarate, contributing 5 carbons from glutamine to the TCA cycle. Now, this is an important process in some cancer cells, making de facto glutamine in these cells an essential rather than a non-essential amino acid. But it is debatable how important that process is in physiologically in vivo conditions. So probably we need to dig deeper to understand this essential process. So this the anaplerotic part of this scheme. So I hope that'll be the end of my introduction. That was a quick crash course on central carbon metabolism. As students, it took us a year to study all this. Now it took us 5 minutes, so that's good. We're making progress. Let's move into leukemic study. This is a work initiated by my lab in Glasgow together with Vigner Helgeson as a group leader at the Leukemia Center and my student at the time, Elodie Kuntz. And what we were trying to find is changes in metabolism of a subcomponent of leukemic, particularly chronic myelogenous leukemia, CML, stem cells. The reason we wanted to study it is, I will explain in a minute, but because this is a component of cells that are resistant to current therapy. So you know that CML is derived from a non-Philadelphia chromosome translocation, the BCR-ABL oncogene, and there is a very effective drug, imatinib, and some other new generation of the tyrosine kinase inhibitors that affect CML quite robustly, in a way, changing the clinical outcome of patients with CML dramatically, as you can see here. So one would ask, why studying CML at all? The reality is that those patients, while the survival rate is amazingly high, they are not cured. Take imatinib off, and there's a very good chance, more than 50%, that the patients will develop the disease again, meaning that we have not eradicated the leukemic cells from these patients. And sometimes resistance is generated, or sometimes patients require to be off the drug, and there's a high risk of developing the disease again. So we wanted to focus on that stem cell component that is probably the component that is resistant to imatinib, and see whether we can identify other vulnerabilities in this component. We work on, by and large, on CD34 positive cells as progenitors, and then CD34, CD38 negative, CD34 positive, CD38 negative cells that are more primitive component of the hematopoietic system, and also the leukemic stem cells for CML. So just to demonstrate what I just told you, we treated CD34 positive patient-derived cells. All the work I'm going to describe here is done with patient-derived cells. We treated with imatinib, and there is a very robust reduction in colony formation or the survival of these cells. However, when we did the long-term colony survival, meaning that first we treated the cells as a bulk, waited for a few weeks, and then placed them in a colony assay, there was no effect, whether treated or untreated, meaning that the long-term colony formation is not affected by imatinib, in line with the ideas that the leukemic stem cells are not affected by the tyrosine kinase inhibitor. So then we took those cells, and we have done a bulk metabolomics just to see what general changes we can see in the steady state levels of different metabolites in a what I call semi-untargeted way. It's not completely untargeted because we do have a list of a few hundred metabolites that we analyzed, but it's a pretty robust way of looking at different classes of metabolites. And the first thing that comes to mind, it's a suggestion, it's not necessarily a mechanism, that we see a decrease in some fatty acids in the CD34 positive component compared to CD34 negative. Of these patients, it was done in 5 or 6 different patients. There is a decrease in fatty acid and an increase in some carnitine linked esterified fatty acids and glycerol 3-phosphate. It suggests potentially that there is an increase in fatty acid oxidation in these cells. These are intermediates of fatty acid oxidation molecules. And there is a decrease in the fatty acid, the free fatty acid components. But that's not approved by all means. So what we do usually in my lab is, in order to validate processes, is we take a kinetic approach of stable isotope tracing. Stable isotopes are, as they're called, they're stable, they're non-radioactive, but they are heavier than the natural abundance isotopes. Mostly carbon-13 is a major friend, which is 1 Dalton larger than the natural abundant carbon-12. So now we can trace atoms, all the carbons in any molecule we wish, as long as it's been enriched to carry carbon-13. So we use palmitate, the fatty acid palmitate, which has 16 carbons, and we use all 16 carbon that are carbon-13 labeled. So this molecule is shifted by 16 Daltons compared to the natural palmitate. But that's less important for us. What is important is now we can trace those carbon into different molecules. And what you see here is this is the process of fatty acid oxidation, end up with acetyl-CoA. If it's derived from palmitate, it will be 2 Dalton larger because it will have 2 carbons derived from palmitate. Then it will enter the TCA cycle, and we'll see the shift in 2 Dalton in every molecule. Of course, there'll be a second round and a third one, and we'll see that shift growing over time with different so-called isotopologues, which is the same molecule with different isotopes. Therefore, they have different masses. Okay, I hope I make it clear. It is important because I'm using this technology along this talk all the time. Now we can trace molecules out of the TCA cycle, for instance, from alpha-ketoglutarate to glutamine production through glutamate. We can actually see the shift in mass in those amino acids. This is important because this is a process that with a mean to an end, if you wish, while the TCA cycle keep on cycling. The same for aspartate. We can look at the transamination of oxaloacetate to aspartate, and we can follow the carbon in aspartate as a readout for palmitate oxidation. This is how it looks once you run the mass spectrometry. You look at the different molecules and their different mass shifts. And what you see here particularly is, again, looking at the amino acids, we see an increase in glutamate derived from palmitate. So every color indicates how many carbons were incorporated into glutamate or any molecule that are derived from palmitate carbon-13. So what you see is the +2, 2 carbon, 3 carbon, 4, etc., 5. Same for aspartate, as you can see here. So indeed, this is now a proof that fatty acid oxidation is accelerated in CD34 positive CML compared to CD34 negative CML. We look now at comparing CD34 positive leukemic versus CD34 normal cells. And again, we see the same tendency. So it is unique to the leukemic stem cell, if you wish, that they have high tendency of fatty acid oxidation. Now, a few years before we made that discovery, Aaron Schimmer from Toronto actually studied leukemic stem cells in AML. And the way he studied it was not mechanistically initially, but actually by a drug screen for FDA-approved drug. And he looked for drugs that specifically deplete AML, CD34 AML cells. And the most effective one was an antibiotic called tetracycline. Tetracycline is a tetracycline-like molecule which affects bacterial protein production because it binds to the bacterial ribosomes. So that's why it's a good antibiotic. However, it has a major side effect, particularly in the heart, the cardiovascular effect. The reason is that it affects the mitochondria. Mitochondria, as you remember, are derived from bacteria, evolutionary speaking, and they're similar and are affected by tetracyclines as well. So what you get is a decrease in the protein production. The few polypeptides that are produced in the mitochondria can be affected by these antibiotics, and that will lead potentially to a lack of effective oxidation and energy crisis. But nevertheless, we manage that toxicity. Obviously, these are very used antibiotics. And we thought maybe to use tetracycline here as a way to inhibit oxidation in leukemic stem cells. So this is what we have done. We took, again, patient-derived cells treated with tetracycline, and we looked at mitochondrial synthesized proteins. They are down, as you can see, while mitochondrial protein that are synthesized in the cytosol, nuclear encoded, are not affected by the antibiotics. Respiration went down, not dramatically, but sufficiently significant that we think that we thought it might be useful to check their effectivity on leukemic stem cells. Doing the palmitate tracing, or actually in this case it's glucose tracing, we could block again the oxidation of the oxidation of carbon in the TCA cycle and the production of glutamate and aspartate from these sources. And then when we looked at the colony formation assay, we could see that there is a nice cooperativity between the antibiotics and imatinib in vitro. But there is no effect on the clonogenicity of normal CD34 cells. It's only the leukemic CD34 cells. So obviously, we wanted to move in vivo to see how well that is working. So we injected patient-derived CD34 positive leukemic cells, waited for them to be engrafted by looking at the human CD45 marker to indicate that we do have human cells within these mice. Then we assigned them to 4 different groups, whether untreated or tetracycline alone, imatinib alone, or both together. And the assumption was that actually, if we look at the leukemic cells, these are the human cells, we will treat the bulk of them with imatinib. These are the CD34 negative cells, but tetracycline will take care of the leukemic component, and hopefully together we will eradicate this tumor. That's exactly what you see here. The combination dramatically decreased the number of leukemic stem cells in these mice. This is actually quantified here, as you can see. Now, the key element, as you remember, was can we actually now put patients maybe in the future, but right now it's mice, off the drug, off the imatinib, and the proof that we eradicated the stem cells will be that there is no recurrent disease after withdrawal of the drug. And here you see in 2 different experiments how imatinib alone did not block the relapse of the leukemia in these mice, but the combination with tigecycline prevented relapse of leukemic cells in these mice, suggesting that we have indeed, to some extent, eradicated the stem cells and hopefully cured now these mice from the disease. So we're kind of thinking, okay, let's move on into clinical trial, and tigecycline is an FDA-approved drug, etc. However, Aaron Schimmer already did that. He tried that recently with AML, following his early discovery with AML. But his conclusion was, unfortunately, that the pharmacokinetic property of tigecycline in human is insufficient to actually engage the target in leukemic cells. So while it's a potent antibiotic, it is not a potent anti-cancer drug in that regard from a pharmacokinetics perspective. It means that you need to develop either more effective or higher doses. Of course, there is the risk of toxicity, and now you start from scratch. Basically, we lose the advantage of the FDA-approved drug and repurposing of this drug. But we all know that there are effective OXPHOS inhibitors developed here at MD Anderson. The IAX 10759. That many of us are using. This is an effective oxidative phosphorylation inhibitor, particularly a complex 1 inhibitor. Clearly, that was an opportunity to explore what I just described in leukemic setups. However, regretfully, that's a recent publication from Team Yap and his team and Mariana. To cut a long story short, the toxicity of that effective inhibitor was probably too high, and there was no recommendation at our current understanding of administration of the drug and the stratification of patients. There is no justification to continue with AML and other patients as well. So this highlights the complexity of targeting metabolism, basically. It's either you get a non-effective compound or not sufficiently effective, or you get a toxic compound. There is a sharp cliff, basically, between effectiveness and toxicity. And this is because targeting metabolism is a tricky business in the clinic. Not impossible, though, and I will highlight at least one follow-up to it that may open an opportunity. And also, there are some examples in different fields in which targeting metabolism can be effective in patients as well. But to follow this, actually, we in that story that I just described, we didn't just trace palmitate, we also traced many other oxidizable substrates, namely glutamine and glucose, as you can see here. And then we traced it into the TCA cycle and into amino acid production, just to read how well they are oxidized in our cells, and particularly in the leukemic stem cell. What we particularly focus on is the stem cell component, the more primitive CD34 positive, CD38 negative cells, as you can see here. And this is glucose tracing here. What you see is a massive increase in the oxidation of glucose in the CD34 positive, CD38 negative of CML compared to the stem cells or normal stem cells. So in a follow-up study done by Vigner Augustin lab with us, they look at the gene expression in alignment with the metabolic alterations that we observed in those cells, again, patient-derived. And what they highlight is that there is an increase both in the citrate production from pyruvate and aspartate production from pyruvate in 2 different manners. That's something I'm going to need to explain. They see the increase in pyruvate dehydrogenase expression and pyruvate carboxylase the non-aplerotic enzymes that I spent some time explaining earlier. What we showed in parallel is a change in the pattern, where you see this process will provide 2 carbons to citrate and then to glutamate or aspartate, while this process will provide 3 carbons from pyruvate. And if you trace glucose, there'll be 3 carbon-13 isotopologues of aspartate, particularly in these cells. And indeed, we see an increase in both the PDH and the PC activity by looking at the 2 carbons and 3 carbons derived from pyruvate by mass spectrometry. However, we don't have effective inhibitors for this. And again, there is a risk of playing around with such key component of metabolism. But what there is available, and actually available in the clinic, is an inhibitor of the mitochondrial pyruvate carrier, okay, that actually slow down the uptake of pyruvate into the mitochondria. And these processes occur in the mitochondria. So what you see here is this drug. This is azemiglicazone, which is a PPAR agonist-like. It's not really a PPAR agonist, but it has an effect on the metabolism of cells, and it was nicely shown to block pyruvate uptake through the mitochondrial pyruvate carrier. And now in a preclinical study, again with CD34 CML cells, we could show that we eliminated the leukemic stem cells, the CD34, CD38 positive, CD38 negative leukemic stem cells, by combining the drug with imatinib, as you can see here. So now this is a drug again used for diabetic patients in the clinic, and we hope to take it to a clinical trial with leukemic patients with CML leukemia. Okay, so that was the first part. That's okay. I'm okay with time. I want to shift gears a little bit, stay with the hematological malignancies, and focus more about the environment now. Unlike solid tumors, which are exposed to hypoxia by definition, lack of nutrients due to growing away from the vasculature system, leukemic cells are not necessarily having that problem of accessibility to nutrients via the circulation. However, some or many of the lymphomas and leukemia metastasize or move to the brain and reside in other subdural area in the brain or in the CSF, in the leptomeningeal regions, particularly for acute lymphoblastic leukemia. What I have here is actually diffuse large B-cell lymphoma. This is how we started this work, which really the CNS relapse is a severe outcome that is an indication to very poor prognosis. So there is a need to identify patients that may relapse in the brain with lymphoma in this case. And there's a list of potential risk factors. 6 of them are more common, and they generate a risk factor combination if you see 1, 2, or all the way up to 6 of these signs in a patient. But the best predictors here is on the kind of on a third of the patient level. Obviously, there is a need to appreciate more what risk factors are available. So we thought that the move from the bone marrow and the circulation and lymph nodes in the case of lymphomas to the brain would require a metabolic shift because of a change of the environment, particularly if the mode of transfer goes through the CSF, the cerebral spinal fluid, that is very poor in nutrients. So we start, and the way to analyze for involvement is also by CSF aspiration and looking for cells in the CSF. So we thought maybe we should look at metabolites in the CSF and see whether we can identify a signature and a risk factor for lymphoma, CNS lymphoma. So together with a group in Paris by Catherine Chivalmont, we used a cohort of 83 patients without CNS lymphoma and 22 with CNS lymphoma at time of diagnosis. And then of those 83 patients, they were followed for 2 years, and 9 out of them developed CNS lymphoma. Now, I should say that all of them were high risk. 6.05, they were considered to be high risk. So you can see how poor this high risk factors poorly define how many will develop CNS lymphoma. We compared these two cohorts, and we're looking for predictors for these ones. Okay, I hope it is clear. I'll cut a long story short. It's unpublished and probably will never be published, because we nicely been able to detect this, but actually none of our predictor, none of our differentiator or classifier, a metabolic classifier, has been able to actually be a good predictor. Okay, so, but nevertheless, it was kind of interesting. These are the top 20 or so metabolites that we identified. Now, as you can see, this was a true untargeted metabolomics where every metabolite is defined solely by its accurate mass and by the retention time on the chromatography. We haven't assigned yet necessarily names to those metabolites. This is something to explore later. These are the top one that we see highly present in the CSF of patients with CNS lymphoma. And what you can see, this is the one of the top one, or the top one. It's not perfect, as you can see, but it has an area under the curve in the raw curve that is the the specificity and the sensitivity curve of 84%. It's not sufficient, but it's a very good start, I should say. What we see here, that this is a lipid. We know that it's a lipid for 2 reasons. It has a high molecular weight compared to the small molecule that we take, and a short run on our chromatography, which is a polar chromatography. So it comes out of the chromatography early. Okay, so we do know that lipids play a role in the CNS and CSF particularly. So this is an interesting observation, which aligns with another work that we have done in parallel, which has been able to reach completion, which is ALL translocation to the central nervous system, to the CNS. This is a collaboration we have done with my clinician partners both in Glasgow, Chris Hazley, and Shai Israeli in Tel Aviv University. Both are leukemia clinicians. And the work was performed by Angela Sapino, a postdoc at the time. Now she's a group leader in Italy. Can't remember where. And Sarah Fernandez, who's now a postdoc in Belgium. So what we were looking for really is a change, metabolic change between acute lymphoblastic leukemia now in the bone marrow and in the circulation in the spleen compared to the brain. And this is what you see. This is the way it typically seen in pediatric ALL, where there is a common presence of leukemic cells in the CSF, in the leptomeningeal region. So we use, again, patient-derived, we started with cell lines, 3 different cell lines of ALL that we injected into mice. And we defined them, we had 3 types of cells. One that has a low tendency, a low tropism to the brain, one that has very high tropism to the brain. We mostly focus on these 2, but we had one that had a kind of an intermediate tropism to be present in the brain. And this is what you see here. This is the bone marrow, and this is the area in the brain that is the submeningeal. When we remove those cells from both sides, those cell lines, and then we subject them to RNA sequencing, bulk RNA sequencing, in order to look for general changes in gene expression pattern, not even focusing on metabolism initially, though obviously I always look at the metabolic angle of such gene expression. And number one feature signature was really a metabolic feature, which was the fatty acid synthesis in the CNS leukemia, those that actually translocated to the CNS. So this is an increase in de novo genes that are involved with de novo synthesis of lipids, fatty acids and lipids. That signify that the cells that go through the cerebral spinal fluid are experiencing starvation for lipids and fatty acids. So we measured those. We actually quantified it in human and in mice. We actually, we managed to get CSF from mice. And we quantified the top fatty acids in the CSF. As you can see here, there's a dramatic decrease in the presence of fatty acids in the CSF, both in human and in mice. And these are the genes that are therefore induced. They are all targets of the transcription factor SREBP, which is induced under lipid starvation conditions. So these are the enzymes that are involved in the citrate conversion to acetyl-CoA in the cytosol, fatty acid synthesis itself that assemble all these acetyl-CoAs to fatty acids together with acetyl-CoA carboxylase. And then also we saw an increase in cholesterol biosynthesis downstream to acetyl-CoA. And downstream to the fatty acid production, we see a massive induction of the gene called steryl-CoA desaturase, which is essential for turning saturated fatty acid, which is the outcome of fatty acid synthesis, de novo fatty acid synthesis, into a monounsaturated fatty acid with one double bond in the middle that make that famous nick in the chain of the fatty acid. So we saw it in the mouse cell lines. Now we looked in patients based on primary samples from CNS versus spleen, comparing patient tumors. We see again the increase in all of these genes, particularly the SCD gene going up quite dramatically. Furthermore, this is using a publicly available data set that look for predictors for CNS involvement from ALL patients, and it shows that the presence of SCD expression in relapse, CNS relapsed tumors, but in bone marrow cells. This was not done in the CNS. There is a high level of expression of SCD, suggesting that actually the presence of SCD is indicative of the ability to metastasize to the brain. So we decided to focus on SCD. We overexpressed it in cells first. You can see here by protein and RNA. More importantly, we look at the function by looking at the ratio between monounsaturated to saturated, whether the 18-0 or 18-1 fatty acids or 16-0 to 16-1 fatty acid. And you can see a change. Sorry, it's the other way, 18-1 to 18-0 and 16-1 to 16-0. And you see a change or an increase in the monounsaturated compared to saturated, indicative of functional SCD in these cells. Now, when we injected it into cells, into mice, we looked at the brain tropism at first sign of CNS leukemia, okay, of these mice. So when we use the high tropic cells, first signs arrive at the time that all mice already has actually CNS leukemia based on our counts, but there is an increase even of those cells that have high tropism to the brain. There is an increase in CNS leukemia. When we looked at the low tropism cells and we sacrificed the mice at first signs was always in the SCD overexpressed cells, and at the time of sacrifice there was no CNS involvement at all in the parental cells. Only the SCD overexpressed cells demonstrated CNS leukemia. So again, indicative of the importance, the essentiality of, not yet the essentiality, but the importance of SCD in this process. To check essentiality, we actually inhibited SCD we used an available tool compound derived from actually UT Southwestern in Dallas, developed that. And now we followed the high tropic 018Z cells into the brain, and you see the decrease in the signal from the brain. Interestingly, what you see here is actually not from the CNS; it's from the bone marrow of the skull. Because when we looked into the CNS, we saw very little cells actually present following the treatment with SCD inhibitor. We have done the same with patient-derived xenografts as well, 4 different patients. And as you can see, 1 of them responded very well, 3 responded okay-ish, some less, some more, but certainly in line with the idea that SCD is essential for CNS leukemia. Again, as is the case for other drugs in the metabolic world, SCD inhibitor hasn't made it yet to the clinic, and there are some indication of potential toxicity, which is now mitigated. We need to be tested whether it's a non-target toxicity or not, but still in process. So we don't have yet a compound that can be taken to the clinic to test for the block of CNS leukemia. However, what we thought about is using that knowledge for something slightly different. So CAR-T, as we all know, is a big player now in cell-based therapy. And in some cases, in some types of CAR-T, there is a major obstacle, which is neurotoxicity, which means that the CAR-T themselves gets to the brain and actually generate probably antigen-specific toxicity in the central nervous system. And that leads to the failure of the treatment, actually stopping the treatment in about 40% of the patients. So that's quite a significant problem from a CAR-T perspective. So what we thought maybe if we now CRISPR out SCD, now we don't need an inhibitor, we can do it ex vivo with the CAR-T. We will not affect their activity in the circulation or in the bone marrow, wherever they need to work, but we will prevent translocation to the brain and neurotoxicity. So this is something, it's an ongoing project. We're working together with Katie Resvani on this project. We only have preliminary data with the leukemic cells where we actually CRISPR'd out SCD, and we blocked the translocation to the brain of the 018Z cells, leukemic cells, without any effect. Actually, there was a slight increase. There is an increase in bone marrow cells following SCD knockout. So now we are trying to do that in CAR-T and see the translocation and neurotoxicity. So that'll be me. I'd like to thank all the people who did the work. It was done in actually 3 different sites. Some of it was finished here, but mostly at my previous lab at the Technion, the Israel Institute of Technology, with Sarah Fernandez leading the leukemia cells, Jonathan the lymphoma work, and before that in parallel in Glasgow as well, the work done by Elodie on the CML stem cells. So thank you very much for your time, and I'll be happy to take questions now.
Thank you very much, Dr. Gottlieb, for these really exciting questions.